It's my pleasure to welcome back a friend and colleague of many years as this afternoon's Grand Round Speaker, Dr. Stuart Hamroff. Dr. Hamroff holds a joint appointment as Professor Emeritus in the Departments of Anesthesiology and Psychology at the U of A. He is the co-founder for the Center for Consciousness Studies at the University of Arizona and currently serves as its director. He has organized several symposia to explore the field of consciousness, has authored or edited books on consciousness, and has numerous articles in peer-reviewed journals and book chapters on the science of consciousness. He has longstanding interest in computation of cytoskeletal microtubules within neurons. This has led him to collaborate with a British physicist, Sir Roger Penrose, in a controversial quantum theory of consciousness. He's also a past recipient of several grants, including one from the Pfizer Institute and the National Science Foundation to study consciousness. On my way over about 15 minutes ago, I ran into one of our fourth-year med students on the elevator, and I said, "Are you coming to Grand Rounds this afternoon?" And his response was, "Are you kidding me? Stuart Hamroff's a celebrity. He was in the movies." So indeed, Dr. Hameroff has obtained celebrity status in more than one area. In terms of consciousness, he has appeared on 2 BBC shows, a PBS show, the Discovery Channel, and in several documentary films. He also has celebrity status as one of the premier basketball players in our interdepartmental UMC tournament that occurs every May. This afternoon, Dr. Hamroff's talk is entitled Recent Developments in the Science of Consciousness. Stuart?
Thank you, John. My basketball celebrity is due to the fact I'm the only one in the league that John can guard. Thank you all for being here. I'm going to talk about some recent developments in the science of consciousness, not so much about my own work, but about what I think has been happening in the last few years that's new and interesting and tells us more about how the brain produces conscious experience. Now, you all make your living on dealing with consciousness and when it goes awry and bad feelings and mental diseases and so forth. And I work as an anesthesiologist where I erase consciousness and bring it back on a daily basis. So we're all kind of in this together. So this first slide shows some different approaches. We take it for granted that when we open our eyes, the world appears before us. And this has been pondered since the Greeks as to how and why this happens. And we know that that the world out there is actually a representation in our head, and we don't really know how accurate it is. And we get into Plato's cave and things like that, some of the philosophy that I'm not going to talk about today. But how do we probe how the brain produces consciousness? Well, here's a neuroscientist literally probing the brain. And by the way, Bing stands for conscious experience, conscious awareness, qualia, phenomenal subjectivity. The robot AI person tries to emulate or simulate consciousness in a machine. The artist tries to capture the essential features of consciousness in art. The physicist ponders the nature of reality, quantum physics and classical physics. The psychiatrist, this is you guys apparently, Freud on his couch, I guess, psychoanalysis and other psychiatric approaches, analyzing consciousness and what goes wrong with it. The meditator looks inward and tries to understand his own conscious awareness. And the philosopher ponders how to best analyze it and what could prove or show what consciousness actually is. So today I'm going to talk about 8 recent developments, relatively recent developments in the science of consciousness. You see them here: default mode, brain networks, self-organizing mobility, and so forth. So I'll go through all of them with varying emphasis on each. I'm going to spend a little time on some and more time on others. So let's start with default mode brain networks. Just curious, how many of you heard of this concept? Okay, a few, but not too many. Basically, before the concept of default mode came along, and still in modern neuroscience and views of consciousness, neuroscientists think of the brain in terms of sensory processing or mental processing the outside world. So we have sensory inputs, and this kind of goes back to Descartes' theater of the content of consciousness being like a stage to be viewed by an audience. So we have sensory inputs come in through the periphery and spinal nerves, cranial nerves, to the thalamus, and then to the cortex, and then feedback from cortex back to thalamus. And this reverberation, or global workspace as it's sometimes called, the thalamocortical system, somehow gives rise to consciousness. And where or who the observer is and why this gives rise to conscious experience is, of course, the mystery. But most views in neuroscience, at least, think of the brain as, think of consciousness as being at least represented in the thalamocortical system with thalamocortical oscillations. And here's an image showing inputs from the spinal cord and cranial nerves to the thalamus, and projections to the cortex, and then back to the thalamus. And this type of feedback has been characterized by Bernie Bars, Gerald Edelman, Tononi, Shanzhou, Dehane, Crick, and Kolk, and a number of others as a global workspace, or thalamocortical oscillations as housing consciousness. And this has been pretty much the standard approach, but it was realized that all of the work that led up to this was based strictly on sensory stimuli and arousal. That is bottom-up inputs from the outside with top-down feedback. For example, PET and fMRI show, in the anesthesia world, show reduced metabolism and blood flow in thalamocortical hierarchical arousal pathways with the onset of anesthesia and loss of consciousness, suggesting a thalamocortical switch. If you turn off the switch, consciousness goes away. And that's been pretty much the party line in anesthesia and a lot of neuroscience, thalamocortical switch. But what about mental states without sensory inputs and arousal? What about task-free, stimulus-independent thought, internally generated states, mind-wandering, episodic memory, meditation, daydreaming? What about when you just kind of let your mind wander and tune off the outside world? Where does that come from? How is that explained, or how is it processed, and how is it characterized or represented in the brain? In 2006, and perhaps earlier, Marcus Rakel and others started to look at this and did a number of very interesting studies and found what he characterized as the brain's dark energy, or default mode networks. And these have particular anatomical regions that you see here in the purple, lateral parietal, posterior cingulate, medial prefrontal, that mediates conscious processing or unconscious processing that occurs without sensory input. So if you're mind wandering, if you're tuning me out and wondering about what you're going to have for dinner tonight, it's these areas in the brain that are probably active. And here they are again in some other work showing these same areas on the median surface and outer surface of the brain. Rakel characterized these in these images, and you can see on the scale bar on the right the colors that these are the brain regions regularly observed to decrease activity during attention-demanding cognitive tasks. So if you're imaging a subject's brain and you have them do something that forces them to be consciously attentive to something, the mind wandering and daydreaming kind of goes away. So these areas in the yellow, for example, are areas that reduce their activity when the subject is forced to do something that requires conscious attention. So therefore, these are presumed to be the regions that are involved in daydreaming, mind wandering, so-called default mode networks. What's going on in the brain when you're not doing something that requires your conscious attention. And here's another view of the axial and coronal imaging showing the default mode network for healthy young subjects. And I was trying to figure out how to recognize this, and I'm not very good at brain imaging. And this configuration with prefrontal and the parietal kept appearing. So I was trying to figure out what it looked like, and to me it kind of looked like Michael Jordan in the Nike ad. So when you see this kind of picture of something like this, sort of looking like Michael Jordan, that's the default mode network. So at least that's their representation in the brain. And sure enough, the default mode network is less active in Alzheimer's patients. So presumably Alzheimer's patients, on top of all their other problems, don't have as much daydreaming or mind wandering as those of us without Alzheimer's. And this work, Jonathan Schooler's group asked subjects to do things like remember their own life, autobiographical memory, again, without sensory inputs, just think about your grandmother, envisioning the future, and the same areas light up. They asked them to think about a theory of mind, how the brain produces consciousness, moral decision-making, all of which don't involve necessarily sensory inputs, but just internal states, and these same areas keep popping up. Rakel and others came up with functions of the default mode network, which include these: episodic memory, introspection, self-referential thought, daydreaming, mind-wandering, envisioning the future, as I said, gauging others' perspective, what do you think about me, context, filling in. This is important. Even when you are processing incoming information, you need context, and what does it mean? And that can be part of the default mode. Thinking about consciousness, meditation, creativity, the Jamesian fringe, something just outside consciousness, the Freudian id, and some aspects of sleep are all attributed to activity in these default mode networks. Now, it turns out that our brains, or consciousness, toggles back and forth between these two types of processing, roughly 50% of the time. And actually, there was an article in today's Huffington Post. I tried to copy the image, but I couldn't get it, which actually studied this. There's some self-help guy who's talking about this, and he said, I think it was 46.9% of the time you're in these default mode networks, not sort of mind-wandering. And he also pointed out that that doesn't necessarily make you happy, but I think that's kind of individual, and if you're going to be depressed and think about depressing things, you're going to be unhappy, but if you're a happy person, so I wouldn't put it that way. But very roughly, 50% of the time you're in conscious mode, and 50% of the time you're mind-wandering, and it toggles back and forth very roughly on the order of 10 seconds. So we have these attention-based sensory processing, task performance, thalamocortical, which has been studied over and over for many years. And then we have the mind-wandering, episodic memory, internally generated states, daydreaming, thinking, just thinking, default mode networks. Again, flipping or toggling back and forth, roughly on the order of 10 seconds. That's just a very rough average. And Rakel calls these anti-correlated networks. And here's a paper in 2005 in PNAS showing the anti-correlated networks. So here you see the Michael Jordan configuration in the blue, and then here you see the same Michael Jordan configuration in the yellow. So between here and here, the subject is going back and forth between paying attention to the here and now, being conscious of what's out there, versus thinking about something else. And it may even be unavoidable. And I know I, for one, do this while I'm driving. I'll be paying attention, and if everything's okay, then I start thinking about something else, and I'm back to looking at the road, back and forth. And apparently we do this pretty much all the time throughout the day. So what does this tell us about consciousness, though? This distinction between default mode networks and sensory-based activities. What does this tell us about consciousness? Well, on the surface, not much, because sensory-based thalamocortical processing, like driving to work, can be either conscious, if you're in intense traffic, you got to pay attention, or non-conscious, so-called autopilot mode or zombie mode, where everything's smooth, you go the same way every day, you know how to get there. All of a sudden, you're at the hospital. How did I get here? Did I drive? I mean, that happens to me all the time. So you're in autopilot mode. So that's sensory-based. Now, default mode network activity can also either be conscious or non-conscious. For example, you could be consciously thinking about your next vacation. You're going to go to the beach, you can go to the mountains, what are you going to do? Or it could be non-conscious. You're thinking, you have a tough day at work, and then somewhere in the brain you're sending impulses to your small bowel to tighten your sphincters or something. So these are non-conscious aspects of the default mode networks. So this suggests that maybe consciousness toggling back and forth is a self-organizing neural process, which is sometimes in the default mode and sometimes in the sensory-based modes. So it suggests that maybe consciousness is something that's a self-organizing process moving around the brain. At least that's what it suggests to me. which brings us to the second point, second recent development, what I call self-organizing mobility. The idea that consciousness isn't, driven by sensory projections, putting something on a screen somewhere in the brain, but it's rather something like an envelope or a soliton or some self-organizing system moving around the brain, and wherever it happens to be, that's what is conscious at that particular moment. So, for example, we have, for both sensory-driven and default mode networks, sensory perception, behavior, all these functions, learning, memory, attention, sleep, language, intelligence, visceral and sexual functions in different anatomical regions, they can be either non-conscious, and philosopher David Chalmers characterized non-conscious processes as the easy problems, or the zombie modes. Zombie meaning being a philosophical entity who might look or act like us without any internal consciousness, or autopilot, and we all know what an autopilot is. For example, in an airplane, the pilot can press a button and the plane flies on autopilot, and the pilot can go take a nap or flirt with the stewardess or have a meal or whatever. And notice that even on autopilot, we can have attention. In the green is something built into the autopilot that highlights something that has a jagged edge or something that might be threatening. And attention can be either conscious or non-conscious. We know this from other work, from Christoph Koch and others. So the autopilot is kind of what's driving the car while we're thinking about our day at work, or while the Wildcats lost the night before or whatever. Now, these same functions at different times can also be conscious. And again, Bing is meant to mean consciousness, so all these functions at other times can be conscious, and but at different times. So different times, what's conscious versus what's not conscious is moving around the brain as we go about, our daily business, roughly moving, very roughly in the order of 10 seconds between default modes and sensory processing. So it seems as though consciousness moves around the brain, and there's some evidence from this, and because we know that gamma synchrony EEG, high frequency EEG in the range of 30 to 90 hertz is the best marker of consciousness, and Sue Pocket and others, Sue Pocket in New Zealand and Walter Freeman in San Francisco have shown zones of gamma synchrony moving around the brain correlating with conscious awareness. Now, gamma synchrony, as you probably know, is 30 to 90 hertz among different brain regions, and from various studies is the best correlate or marker of consciousness. One very interesting study on meditating monks that was done in Rich Davidson's lab a few years ago. The monks were chosen by the Dalai Lama for their expertise in meditation, and you might say that they had a higher state of consciousness, a more enlightened state, or more intense consciousness, and it turns out, had the highest gamma synchrony ever recorded in humans, except for seizures, which may not be gamma, but the highest amplitude, frequency, and coherence in the gamma range. And the control group were college kids who were taught to meditate over a couple days, and they had a little Bing, as you can see here, and some gamma, but nothing like the monks, who went from, normally we're about 40 hertz, 40 conscious moments per second, you might say, whereas the monks were between 80 and 100, higher amplitude, higher degree of coherence. And even before they started meditating, their gamma synchrony was higher. So years of training apparently can cause the brain to change so that they can have more gamma synchrony than the rest of us. So that suggests that maybe it's a good thing to do, although I don't do it myself. So how might this happen? Now I'm going to digress a little bit into my own work, because I think here's one way to explain how consciousness can move around the brain. And we have two types of synapses in the brain. We have chemical synapses with the neurotransmitters and neuromodulators, and this is how a lot of the psychiatric drugs work by altering synaptic plasticity or synaptic sensitivity and so forth. But we also have these electrical synapses, or gap junctions, usually between dendrites, and these synchronize neighboring neuronal membranes so that they not necessarily fire synchronously, because these are dendritic potentials, but they depolarize synchronously, and this gives rise to gamma synchrony. So this is a sort of sideways connection, gap junctions, dendritic, as opposed to axonal dendritic or axonal somatic, which is kind of a serial. This is more parallel. And we know, so here's a circuit of sideways connected neurons, and there's bang and there's coherence throughout the entire system. So this type of connection, gap junction connections between dendrites and also involving glia, and here's some glia, because they have gap junction, they're part of this type of system. Synchronous gamma activity in these types of networks seem to correlate with consciousness, which is why I have Bing there, because we know that that correlates with consciousness. So if we have a network of toy neurons with inputs on the left and outputs on the right and up top and the bottom, and just have this is a conventional type of neural network based on chemical synapses. But then we have a gap junction open here. There's synchrony there, and it grows and moves around, and Bing goes with it. So this shows how gap junction connections can form an envelope of synchronized neurons literally moving around the brain. And I think that, personally, I think that is how consciousness is able to move around the brain through these sideways connections while everything else is ongoing. So this shows, I showed this before, consciousness moving around the brain, and the idea that the gap junction connections are responsible for this envelope, mobile envelope as an agent of consciousness moving through the network. I call it the conscious pilot. Dendritic synchrony moves through the brain to mediate consciousness, which I published early this year. And so the conscious pilot, and here's one example, if we go back to the airplane analogy, he might put the plane on autopilot and go flirt with a stewardess or whatever, and just like we might be thinking about something else while our autopilot is driving our car to work. Now, two examples of gamma synchrony mediated by gap junction mediating consciousness in the brain are these. One is olfaction, and it's interesting that conscious smell doesn't go through thalamus. I think it's the only sensory system that doesn't require thalamus. It goes right into the olfactory bulb. And these two papers show that conscious smell correlates with gap junction mediated gamma synchrony in olfactory bulb cortex. And by blocking they showed this by blocking the gap junctions and taking away the sensation. And now another example of this is feelings of pleasure and reward, which we know are correlated with dopaminergic activity in nucleus accumbens and prefrontal cortex and ventral tegmentum. And it turns out that that gap junction mediated gamma synchrony in these areas mediate pleasure. Now, they're dopaminergic systems, so these are still active, still involve dopamine, but they're not always conscious. So when these dopaminergic systems get sort of taken over or involved in a gap junction mediated envelope of synchrony, we become conscious. So, and as you know, this is kind of the final common pathway of pleasure, whether it be food, sex, thrills from skydiving, or whatever, are mediated through this system, which involves dopamine, but also involves a gap junction mediated gamma synchrony. Okay, so that's self-organizing mobility. The next topic is scale-free brain dynamics, and I just learned about this in the last year. And Marcus Rakel, who developed the default mode networks, is also involved in this. So scale-free dynamics, what do I mean by that? It's something like a fractal or a hologram. And I'm not going to get into the math because it's a bit above me, but it means self-similarity across various scales. You know what a fractal is. You sort of know what a hologram is. It means that the frequencies, the size in terms of amplitude, and the energy at various scales is similar. So if you measure at a low level, you get a signal. If you measure at a higher level, you get the same information, just at higher amplitude, different frequencies, and so forth. It's also found in holograms and is known in nonlinear dynamics and chaos theory as self-organized criticality, 1 over F noise, 1 over the frequency, or pink noise. And it's found in many, many systems in nature and also in the brain. And for example, here are a few references. Again, here's Rakel. So it's found in EEG microstates. It's found in wake-sleep cycles. It's found in eye movements and saccadic eye movements on animal or us. We'll look look at the whole field and then zoom in and use the same sort of tracking. So it's a fractal type process. And it's actually, when you think about it, it's natural because a neuron embedded in an intact brain, unlike an isolated neuron, participates in networks at various spatial resolutions. We have so-called local interactions or near, small world interactions, but also large world interactions. So any neuron is in multiple networks at different scales, and different size scales and multiple time scales, giving rise to the possibility of a scale-independent memory preserving mechanism. So just one slide of the mass showing log of frequency versus log of power, and over different orders of magnitude, it's more or less linear. Obviously, it's not totally linear, but it's more or less linear over different scales, suggesting that the scale-free dynamics are the same. And this has been found in EEG, in interspike intervals, saccadic eye movements, neuronal architecture, and actual structure of the brain, with time scales from tens of seconds very long to thousands of hertz, very, very small. So the same type of dynamics and signal is found at different levels. And this harkens back to some theories that go back early in the century from Carl Ashley and more recently Carl Pribram, that consciousness and memory are sort of a hologram or stored or processed somewhat like a hologram or a fractal. So I suspect we're going to be hearing and seeing a lot more about that. The next category is electromagnetic field effects in the brain. I'm not going to spend a whole lot of time on this. I will mention this conference that we're organizing. John mentioned that our Center for Consciousness Studies organizes conferences every 2 years in Tucson and an odd number of years elsewhere in the world. And this year it's going to be in Stockholm, Sweden. And I show this slide at this time because we're going to have a session on this topic of brain fields or electromagnetic fields in the brain. And the background for this is that these two people, John Joe McFadden and Sue Pocket, for many, many years have argued that consciousness is actually the field in the brain, the electromagnetic field. And it's very complex. It's determined by the neural activities. And I've always been opposed to this, and I've argued with them and said, well, you know, there's an electromagnetic field everywhere. How does the brain differ ontologically? Why wouldn't you say that the electric field in a computer or a television was conscious? It's just a different type of, and they have arguments around it, but I never really took it seriously, nor did too many other people. But then recently, this guy, David McCormick, and many other people have shown that the field itself actually has feedback on neural activity. So it's kind of like the Indian rope trick. You know, the guy throws the rope up and climbs up. The brain generates a field, electromagnetic fields from neural activities. That field then feeds back and influences neural activities. And I think this gives credence to the idea that the electromagnetic field in the brain is what is conscious. I still don't believe it, but I think it's a reasonable hypothesis, and we'll have a session on this in the Sweden conference, that the brain-generated electromagnetic field providing feedback bolsters these theories of consciousness. And that leads to the next category, which is transcranial therapies. And we're having another session on that. And I know that you guys are familiar with transcranial magnetic stimulation for depression. And Eric Wasserman from NIH will speak about that at the Stockholm meeting. And we know a large magnetic field near the brain can block neural activity near it, but it apparently can also have beneficial effects on depression and perhaps other things. I asked John about it, and he wasn't too impressed with it, and I think people have different opinions, but it does do something. and it's sort of a new area. Now, along with transcranial magnetic stimulation, Alan Snyder is going to talk about transcranial electrical fields for memory enhancement, and he's actually using electrical stimulation of the brain to uncover what he calls savant capabilities within normal people. He thinks that we all have enormous memory potential and intellectual capacities that are blocked, and if you sort of inhibit the inhibitory processes, you can bring out what's underneath. But he's also an expert in transcranial magnetic stimulation. So we'll be hearing about that. And the final category I find the most interesting, and it's going to be discussed by a guy from Arizona State named Jamie Tyler, transcranial ultrasound for mental disorders. And so let me just talk about that. This I got off the web a while ago from a military site announcing a grant to William Jamie Tyler at ASU using ultrasound activity to control brain activity. Now, it says remote control, but they're actually talking about helmets because ultrasound doesn't go through air too well. You have to use it with gel to get to the skin, and then it actually goes through the skull with some pretty good efficacy. And so it's not remote in the sense of somewhere out there. It's actually right on the skull surface. And he got a lot of money from the military to study this for cognitive enhancement, to allow soldiers to have peak performance, to improve their abilities in the battlefield, but also for treatment of traumatic brain injury and various things. And Tyler has a spin-off company to make ultrasound devices. Here's some helmets and stuff with ultrasound transducers that he would put on the surface of the brain, and they're promising or suggesting neurological or psychiatric intervention, navigational commands assisting, alertness, wakefulness, behavioral reinforcement, anxiety, stress reduction, cognitive enhancement, pain intervention, traumatic brain injury protection. Now, I should back up and say people were noticing behavioral effects in animals of applying ultrasound, but they weren't sure whether the signal was actually getting into the brain. So what Tyler did was, in animals, mice actually, was put electrodes into the brain of these mice, let them recover so they were awake and walking around or whatever they do, and they could record from the brain. And he then did transcranial ultrasound and found not only behavioral changes, but also electrophysiological changes from the applied ultrasound. So the ultrasound actually does something physiologically, does get into the skull, through the skull, into the brain, and has some efficacy in doing something. And they also are suggesting that multiple transducers or multiple inputs of ultrasound. So, for example, you could have it at two different places, and where they intersect in the brain would be positive reinforcement, constructive interference, or positive feedback, and you could focus it deep within the brain. For example, maybe the nucleus accumbens. I don't know if many of you remember the Woody Allen movie about the orgasmatron, but something like that might even be possible to focus in particular areas, to stimulate particular areas deep within the brain. Our group at the University of Arizona, my colleague John Badal and I, his wife is a psychiatrist in your department, at Keno anyway, and we've put in a protocol to a human subjects committee to try transcranial ultrasound for chronic pain therapy. And we're skipping all the mice and rat work because Tyler's done that for us. And we use ultrasound every day in anesthesiology for nerve blocks, in imaging. These are the exact same devices that we use to image nerves and blood vessels to do central lines or to do nerve blocks. It's what is used in OB to examine the fetus. The ultrasound goes in, it's reflected back, and you get an image. And it's even approved, the device that we use every day is approved for transcranial imaging. But that was years ago, and now with functional imaging, the various types of imaging we have, it's kind of it's become obsolete, but it's still approved. So what we've done is taken the device that we use for brain imaging, which we use, for nerve block and central line placement, and we use every day on dozens of patients a day, and using that for transcranial stimulation. And our first approach is going to be for chronic pain. So what does it do? Well, we experiment on ourselves, and I can tell you it has a very interesting and very pleasant effect on the brain. So I'll just leave it at that, and we'll see what happens with our chronic pain study. And if that works, we hope to try it for memory. Now, I should back up and say that Tyler, the other people working in this field don't really have a good mechanism, a good explanation for what the ultrasound is doing. They say, well, it's doing something to the membrane, something about synapses. I don't think they have a good explanation. Now, I've been interested, as John mentioned, in microtubules for 35 years inside neurons. And it turns out that microtubules have coherent resonances in the ultrasound region, that is megahertz, 10 to the 7th cycles per second. And if that's electromagnetic, it's in the radio frequency, and you don't want that. But in mechanical vibration, it's ultrasound. So it's too fast to hear, but it's just mechanical oscillations in the range of roughly megahertz, 10 to the 7th cycles per second. So that's what got me interested in it, because I'm thinking, well, if we're revving up the microtubules, that's going to enhance synaptic plasticity, turnover, and that can improve learning. And since Alzheimer's involves defects in microtubules, it could possibly work for Alzheimer's. So we hope to try it for memory. After that, for things like as mundane as nausea and vomiting with surgery. And I've mentioned to a number of people that it could be a valid, very interesting technique for psychiatric intervention. And as I said, Tyler, for one, has, you can see the list of things that he's suggesting it for, and he has a spin-off company that's looking for investors and whatnot. But as I said, we're skipping the rats and going right to people. And so we'll let you know how it works out with chronic pain, and if that works, we'll go on to memory. Our next protocol will be for normal people, have them do a memory test, be exposed to either the ultrasound or control, and you can't really tell that it's on. When you put it on, you don't hear it, you don't feel it, you don't feel a thing, and then you put it down, and about 10 seconds later, you start to get a sort of a buzz, actually. So we'll see what happens with that. And here's a microtubule, which has resonant frequency in the megahertz range. Okay, so number 6 is molecular memory and cognition. Actually, I was here a couple years ago and was researching about memory because a guy was trying to erase memories for post-traumatic stress syndrome. And this led me into a paper that I'll get to in a second. But when you think about single-cell paramecium can learn, avoid predators, find food, mate, and have sex. It doesn't have any synapses. How does it do it? And here's two paramecium mating, actually. It's kind of an X-rated slide, along with the nucleus accumbens slides. It uses its microtubules, both on the surface in the cilia and internally. And we also have the same microtubules inside neurons, which regulate synapses. Here they are. So here's 2 synapses. This is a dendritic spine synapse for glutamate. Here's an acetylcholine synapse right on the dendritic shaft. The microtubules form networks, information processing system within. And I've always thought that I got interested in these in medical school back in the 70s, that they might be processing information, it might be molecular computers. And we know that in Alzheimer's disease, there's 2 lesions. There's the amyloid plaques, which for some reason get all the money, and the neurofibrillary tangles inside, which I think cause the cognitive defects. And if we look at an Alzheimer's neuron, here's a diseased neuron. It's all kind of crinkled up, and its microtubules are literally disintegrating because this protein, tau, has been hyperphosphorylated and fallen off, and the microtubule just kind of disintegrates. And that leads to cognitive defects in memory and eventually consciousness, but first certainly memory. So what does tau do? You know, tau is always, when I read about it was always, well, tau kind of knits the microtubule and keeps it from falling apart. Well, recently we've shown that tau actually has an active role, you know, when the neuron's working properly, before the before Alzheimer's sets in. So tau, under normal circumstances, literally acts as a traffic signal for motor protein transport of synaptic material. So here's a dendrite here. Let's say this is the axon here. And we're zooming in at one microtubule in this dendrite. So out here is a synapse that needs, I don't know, dopamine decarboxylase or something, which is being transported along it by these motor proteins called dynein. These strange-looking things that literally prance along the microtubules quite rapidly to get to the synapse. So the question is, how do these dyneins know when to release their cargo? How do they know if you have synaptic plasticity and a particular synapse needs an enzyme or needs a receptor precursor or needs whatever due to synaptic plasticity and local learning? How does the cargo know where to get off? So it turns out that tau is kind of a traffic signal, and here, for example, has told this kinesin to jump ship and deliver its cargo to the synapse that's just above it on the membrane. And so then the question is, well, how does the tau, so then the placement of the tau specifically on the microtubule will tell the motor protein where to get off and where to deliver the cargo. So that means it's either the tau is very intelligent, which seems unlikely, or there's information encoded in the microtubule itself for where the tau should bind. And I think the latter is much more logical. Which brings us to memory. And this is something that I looked at when I was here, I think it was a couple of years ago, about memory and post-traumatic stress syndrome. So we know that in long-term potentiation, which is the model for memory, calcium comes in and activates an enzyme called CAMK2, calcium calmodulin kinase 2, which is this snowflake-shaped enzyme, which then binds to the microtubules and is transported throughout the neuron, and within seconds after activation, after the LTP, it's distributed widely throughout the dendritic arbor. Well, this CAMK2 actually looks something like this. Here it is in its unactivated form. Here we're looking on top of it. It's a hexagonal snowflake-shaped molecule. Here we're looking on the side. And when it's activated and gets phosphorylated by calcium coming in, so in a learning paradigm, it transforms, kind of like Transformers in the movies, where these legs come up into this insect-like, I don't know what to call it, a nanopoodle, somebody calls it, with 6 legs down and 6 legs up. And each of the legs, the kinases they are actually, can phosphorylate a substrate, and that stores memory. So we don't know what that substrate is, but we know that when CAMK2 phosphorylates something, memory gets stored. So the question is, what is CAMK2 phosphorylating, which is probably the same answer as where is memory stored, or at least short-term memory. Well, being a microtubule fanatic, you might expect that we looked at microtubules as a possible recipient of phosphorylation encoding of memory by CAMK2. So here's a microtubule. It's hollow. Here it is on end. It has these C-termini hairs coming off of it. And in the blue and the cyan are the phosphorylation sites on tubulin, which can get phosphorylated by CAMK2 or any phosphorylating agent. These are different scales here. So the question is, how does CAMK2 relate geometrically, structurally, size-wise to microtubules? And the answer is perfectly. And so here's CAMK2 with, say, the top taken off so we can see the 6 kinases. This is all 10 nanometer scale bar. Here's a lattice and a B lattice of a microtubule. And here we have the CAMK2 overlying the lattice, and we see that the 6 kinases match up perfectly, and in the B lattice also in 2 different orientations. So it looks like it, and we know that CAMK2 is transported or transports itself along microtubules, and so it could be something like this. I showed you the kinine and kinesins and dyneins before, and so CAMK2 is another microtubule-associated enzyme or holoenzyme that can literally match up with the lattice and deposit information by phosphorylation. And here we show how the phosphorylation can occur literally down to the amino acid level, 2 different possibilities. And the bottom line is that if we look at a little piece of a microtubule lattice and assume that the CAMK2 can have 6 bits of information, because each kinase can phosphorylate or not. And an ordered array of bits is called a byte, that we could have bytes of up to, we only show 6 here, but there can be up to 62 to the 6 or 64 possible bits per byte. Here, if we make some other assumptions of trinary states, something like 500, and in this case, 5,000 bits per byte for one CAMK2 encoding information on one little region of a microtubule. So this is the potential information storage capacity and encoding of CAMK2 on a microtubules is enormous. And this paper just came out in the Journal of Integrative Neuroscience. And if you'd like a reprint, send me an email and I'll send it to you. So this speaks to the idea that, the more general idea that microtubules may process information. But that's another story. So I want to wrap up with two somewhat more controversial, well, not that those weren't controversial, but controversial topics that I think are very, very interesting. One is end-of-life brain activity. And, we've heard for many, many years that people who have cardiac arrests and then are resuscitated, revived, a certain percentage of them, actually about 17%, describe what's called the near-death experience, where they see a white light, a tunnel, and in some cases actually leave their body. Here's somebody waving goodbye to their body. And if they're revived, if they're resuscitated, they come back and they say, I saw the white light, I saw my dead relatives, et cetera, et cetera. And, we tend to think, yeah, right, it's just some kind of hallucination, you were hypoxic, and there's all kinds of skeptical replies to this. But none of those skeptical replies really explain it, because, I've seen a lot of hypoxic patients, and they don't have this clarity and serenity. They're agitated, confused, and scared. And these patients are serene and calm. So anyway, recently, two groups have done processed EEG of patients as they die. And one group was a palliative care study done by a guy named Chawla at George Washington University, and another was done by this group at Virginia Mason in Seattle for patients who are severely brain damaged or brain dead for organ donation after cardiac death. These are patients who are severely brain damaged, but for one reason or another, they can't declare them brain dead. So the family wants to donate the organs. The patients are obviously hopeless. They take them to the recovery room. They let them die. And as soon as they're literally dead, they're pronounced, their heart has stopped, they take them to the operating room and take their organs. So I'm not sure why, but they put these processed EEG monitors on them. These are monitors we use in anesthesia to give us a sort of an idiot number from 0 to 100 of how deep or light the patient is. So it's derived from EEG. It's proprietary exactly how they come up with the number. It involves gamma synchrony, but there's some other things to it that they won't tell us. But the bottom line is that you have a number from 0 to 100 over here. And BIS is one, the bispectral analysis from Aspect Medical. Another one is the SED line that we have here. And they both use 0 to 100. And both of them say, 80 to 100 is where normal awake people are. We want our anesthetized patients between 40 and 60. If they're between 40 and 60, they won't be awake, they won't remember, and they won't sue us for awareness. And if they're below that, they're brain damaged for whatever reason. So here's one. Chawla published a study of 7 patients, and Aoyang et al. Published 3 patients. So 10 patients in all. All 10 at the time when the heart was stopped or there was no blood pressure. So they're basically clinically dead. This number indicating level of consciousness or level of awareness, which is already well below the level of awareness, down near 0, the heart stops, and all 10 patients had this burst of activity up to above 80. This particular one lasted for like 15 minutes or 20 minutes. They lasted anywhere from 90 seconds to, in this case, 20 minutes. And in one study, they looked at the raw data, and it was gamma synchrony EEG. So what this is, we have no idea. I think Chawla, suggested that this could be the neural correlate of the near-death experience, of the white light, of the maybe even out-of-body experience and so forth. Of course, these patients died, so we don't know. He's going to do more studies, and maybe they will be revived and will know if they had the white light or experience. We don't really know. But it's a very interesting phenomenon, and again, something that will be discussed at the Stockholm meeting that we may hear, and we may be hearing more about this. It's going to hit the lay press, I'm pretty sure, one way or the other. At the risk of you guys thinking that I need to see a
not
psychiatrist,
necessarily a supernatural.
So can you
This
tell us
was,
a little bit about how you think of consciousness?
I think most people would see the brain as a computer of 100 They
billion
asked me about this,
simple
about
switches.
a
Each
case of
neuron being a
reincarnation.
switch.
It can fire or not fire. But neurons as a cell are incredibly complicated. And I think we have to go down a level below the level of neurons, inside the neurons, to the level of the microtubules. The blood stops flowing and the oxygen isn't being delivered and the brain starts to lose it, die essentially. The quantum information that is consciousness, including memories, isn't necessarily lost, but can dissipate at large. So it's possible that consciousness, including memories, can exist outside the brain, outside the body. And in the case of near-death and out-of-body experience, would go back inside. If the patient died, then perhaps could be drawn back into another organism, another set of microtubules in a zygote or an embryo.
He's laid out a plausible theory about how
This is from a show that's
consciousness
going to be on the Oprah
exists
Winfrey
independent
Network,
of the
I think
brain
after the first of
after
the
life.
year,
So do you
called
believe it's
Mystery
possible you
Detectives,
could be
or Miracle
reincarnated?
Detectives. These two guys,
It's
these two go
possible, I
around
hope
and
so.
investigate.
We'll find out.
This was a case of reincarnation. A kid who had memories of a World War II pilot described details that there was no way that it's hard to understand how he could know that. So I just give a plausibility argument.
I would say no. But I think it's
The
possible.
lady's a
I learned a lot
neuroscientist. She's the
about how
hardcore
Dr.
skeptic, and
Hameroff
the
thinks
guy is kind
in
of the
terms
woo-woo
consciousness
new age
and
guy.
its representation in the brain. But what he's presented to us is a theory. He doesn't have direct evidence, and only time will tell whether his theory is correct.
Thank you. So I just want to wrap up a little bit about quantum, and I'll just, this is a whole lecture in itself, but let me just say a couple things. That quantum is, reality is described by two distinct sets of laws, classical and quantum, which is small, but not necessarily small. It can happen at large scales. And four features are superposition, quantum entanglement over distance, quantum coherence, and uncertainty. And this is the model that Penrose and I developed. And basically, it involves proteins switching states due to electron resonances in nonpolar rings. And these same nonpolar rings are found in all psychoactive drugs, including the psychiatric drugs, as well as the psychedelic drugs like DMT shown here. So, and studies have shown that the more electron resonance donated from the drug to the receptor, the more hallucinogenic the compound. So the basic idea is that quantum, you have multiple coexisting possibilities where classical is definite, deep interconnectedness versus isolated, timelessness, non-locality, possibility of spirituality versus material. And I think from the psychiatry standpoint that the quantum quantum information may be involved in subconscious dreams and altered states. And you could, conjecture maybe schizophrenia or something where you're too much in the quantum and you're too much like in a dream world and reality isn't what it should be. So I think consciousness may be a process on the edge between the quantum and classical worlds. So just to close, let me just again mention the Sweden conference, and my assistant Abby Behar Montefiore is here, who runs the Center for Conscious Studies, has been doing a fantastic job organizing the conference. And then we have next year it'll be back in Tucson. So I'll just close and give you this slide of the summary of the what I think are the 8 new recent developments in the science of consciousness. Thank you very much. Dr.